Yeah, is that on? Yeah. Yeah. Morning, everyone. Morning. Welcome to the Soils Tent. Thanks for coming. Lovely morning this morning compared to yesterday. It was a bit cold, but it's nice to see the sunshine today. My name's Anne Bhogal. I'm a soil scientist at ADAS. And it's going to be a bit of a double act this morning between myself and John Elphinstone from Fera, a plant pathologist at Fera. And we're going to be looking at, the title is Measuring and Optimising Soil Management Using a Soil Health Scorecard Approach. I'll cover that bit. And then John is going to come up and look at some of the more emerging molecular techniques for assessing soil health. So I'm preaching to the converted here. You all recognise that soil is your greatest asset. This Groundswell is fantastic in promoting healthy soils, not only from a crop production point of view, but from the point of view that actually soils are incredibly important for the wider environment in terms of regulating water flow, atmospheric gases, source of biodiversity, and also supporting infrastructure and various cultural things that we do. So fundamental. But certainly for agricultural soils, there are a number of key threats to their condition. I've identified three. The first, soil compaction. I carried out a bit of a review of a number of surveys that have been carried out over the last 10 years or so of agricultural soils in the UK. These looked at the structural condition, so visual assessment of soil structure, ranging from the southwest of England right up to Scotland. And from horticulture to arable to grassland. And pretty much the story was the same. Around about 40 to 60% of the soils were what would be classed as moderate conditions. So there's some evidence of compaction. And anything up to 30% are severely degraded. So have a very strong plough or cow pan and showing evidence of surface erosion. And erosion is that next threat to soils, loss of topsoil. Here's just some numbers. Background erosion rates around about 0.1 to 0.5 tonnes per hectare per year in the UK. Compare that to cultivated land, anything up to 10 tonnes per hectare per year can be lost. And then if you leave that land fallow, that cultivated land fallow, you can get massive erosion losses in the forms of gullies and channels. Compare that to formation of soils, you can see that we're going in the wrong direction in terms of soil loss. And then perhaps underpinning all those processes is the loss of soil organic matter that we've seen over the last few decades. This is just a chart of the Countryside Survey in 2007 compared to its previous survey about 5 years earlier. And the change in organic matter that had happened over that period under different crop types, land use types. So we've got the more natural habitats over here, which actually showing they're quite variable, but on the plus side. And the only significant change with the stars is in this crops and weeds, so the arable. A loss of organic matter measured in that. And so that obviously has consequences for crop production. Water quality and greenhouse gas emissions. And so soil organic matter is really the underpinning thing behind those other processes. It's the stuff that gives us the resilience to compaction, to erosion, largely because it fuels all the biology in the soil. And that biology in turn cycles nutrients, helps with soil structure. The soil structure then improves water holding. So you can see that the central pin to soil health is soil organic matter and a key indicator of the overall health of the soil. So with that in mind, obviously recognising soil health is important, we then face with lots of questions. The key one is, how on earth do I know if my soil is healthy? What do I need to assess? And when I know what to assess, how on earth do I benchmark? How do I interpret what I see? And then what management practices should I be putting in place when I know I understand what I see? And then there's lots of questions about soil biology in particular. I've said that processes are fueled by soil biology, but actually that's probably the one aspect of soils that we don't know as much about. So in response to that, AHDB and BBRO have set up this 5-year partnership of research and knowledge exchange aimed specifically to improve our understanding of soil biology and soil health. Both extend the research but also come up with practical measures that we can use on farm. So as I say, it's a 5-year partnership led by 5 research organisations, but also involving a number of industrial partners. And a key aim of it is to develop and validate indicators of soil biology and soil health, using both research trials, but also with farmers. And farmers are key in this process of learning and exchange. But a key aspect is that we're building on what we already know. So we don't want to start at square one. So what do we know? So we spent a first year just looking at the literature, looking at what's going on out there, and what do we know? Well, soils are complex. Take this example. This is the example of the function of nutrient supply. So when you think about nutrient supply in the soil, you need to think about the soil physics. So what's my soil texture, what's my minerals, how aerated is my soil. Then you also need to think about the chemistry, what are the nutrients there, what's the pH. The biology, so what recycling of organic matter is going on. But also the plant, how is the plant rooting, what are its root characteristics, its uptake efficiency. So those are the things we need to look at, but they are all moderated by where you are in the country, the climate, the temperature, the rainfall. And then on top of that, you're manipulating it with your nutrient inputs, your manures, your fertilisers. So there's a huge amount to think about when we come to think about soil health. So just taking that a bit further, looking at the soil, as I say, key is to understand the chemistry, the physics and the biology, all three in an integrated way. So the properties can be divided into those which are inherent. So those are what you've got to work with. They're the limits of that soil habitat. So they would be the texture, the depth, for example. And then you've got more dynamic properties, which is basically the nature and the composition of that soil habitat. So the organic matter content, the nutrient content, what's feeding the soil. So those are things actually that we could potentially be manipulating. And then on top of that, you've also got the influence of crop. What are you growing? What residues are going back in? How do they root? What interactions are going on with those roots? Are there positive associations? Is there negative associations going on? And what have I got in my seed bank? So taking that integrated approach for looking at soils, we also then take that approach for looking about how we might manage those soils. So the biology, the physics, the chemistry. So examples might make sure you feed the soil regularly, only move soil when you have to. On the chemistry, maintain the optimum pH and check your nutrient status. And on the physics, think about your textures and when you go on and actually do your operations and cultivations times, so you're not doing them when it's perhaps too wet and cause more damage. So in terms of the practices that you might do linked to that, in terms of feeding the biology, it's things like organic material additions, it's introducing grass lays, cover cropping, reducing your tillage, diversifying your rotations. But also, as I say, we need to be thinking about when you traffic the soil, when you go on the soil, making sure the timing of your operations is correct and your type of operations is appropriate. And then also perhaps think about drainage. So if we go back to soil health assessment, as I say, part of the programme is to come up with some easy tools with which to assess your soils. So again, we need to think about physics, chemistry and biology in an integrated way. So that will involve not just taking a sample and sending it off to a lab. It means going out with your spade at the same time and having a look. So physical examination coupled with sampling and sending samples off to the lab. And we're saying do this, it doesn't have to be done every year in every field, on a rotational basis. Pick a crop and do it at the same time of year so that you can actually, over the course of, say, 4-year cycle, track the changes that you might see in those soil properties. So what do you measure? when you come to take your soil sample or look at what you see. There's been a huge amount of work developing indicators, and there's hundreds and hundreds of indicators out there. DEFRA, in a part of a soil monitoring programme, they've funded several projects looking at different indicators. So physical properties, there's a report that looked at 42 different physical properties, and they suggested there might be 7 that would be useful. Biological indicators, a huge piece of it, 183 candidate targets that you could measure, of which they suggested 21 might be useful. And the EA, from an environmental perspective, had done the same. They reviewed 67 different indicators and came up with 11. And from that, we can see that no one indicator will do the job. And there'll be some indicators that are really only useful for the research environment to understand soil processes, whereas there's others which would be more for practical use on farm. So with that in mind, we did a similar exercise, having looked at all this, and we reviewed about 45 different indicators that could be practically used on farm. Soil physics, recognising that we need to look at soil physics, chemistry and biology, and we wanted to adopt a sort of scorecard approach. So those indicators, we needed to be able to interpret what we see. So a bit like a school report that I might get from my son, who says, he's great at maths, he's absolutely appalling at English, and he needs to work harder at science. That kind of thing, so that you can see and track, okay, well, my soil chemical properties are fine, but actually I've got a bit of compaction there that I need to address. So you can see where you might tackle the issues that you find. So these are the indicators that we came up. We came as physical properties. We had 17 candidates, and we scored them basically. We evaluated on how relevant they were, whether they were easy to use, their cost effectiveness, and whether they had a potential for an interpretation scheme in place. So for physics, a visual assessment, get your spade out and have a look and see what you see. Penetration resistance to measure soil compaction, and bulk density. For chemistry, pH, nutrients and soil organic matter. And for biology, earthworm numbers, respiration and microbial biomass. So those are the ones that we're evaluating. They may not be our top indicators when we come to the end of the programme, but those are the ones we're starting with. So as I say, it's important that we might be able to benchmark what we see so we can actually interpret what we see. So where are we at in terms of benchmarking? So these green ones are quite established in terms of benchmarks. So I've given them some ticks, pH, routine nutrients. We've got some information on bulk density and penetration resistance and VES. Soil organic matter, we've developed an approach to testing that. Earthworm numbers, but things like respiration and microbial biomass, I put partial. We've got data from international data, but is that appropriate to UK conditions? And then I've put these new indicators, because there's a second aspect of the work, is to try and extend our understanding of soil biology. So at the research sites where we're testing the scorecard, we're also undertaking a whole suite of other measurements to try and understand the drivers of soil biology and what a potential indicator might be. So things like potentially mineralisable N, DNA techniques, which John will go into, but also counting nematodes and mesofauna to try and understand, are there some indicators there that we could potentially be using? So if I just spend a little bit of time about how we're benchmarking it. So for the routine nutrients and pH, that's basically using RB209 as our guide, looking at the index system. For visual soil assessment, we're using SRUC guidance, and I'll go into that in a minute, but we're scoring what I call the limiting layer. So the worst layer in that topsoil, we give it a score. And soil organic matter, we're comparing those levels to what is typical for where you are in the country, your soil type and your climatic region. And I'll go into that in a bit as well. And the way we're scoring it is a very simple traffic light system. So if you're green, that means you're within your range. Everything's fine. You don't need to worry about it. Amber means actually you're sort of borderline. Maybe that means you need to go out more regularly to sample, perhaps not on the 4. Go and have a look next year, or go back again now and have a little look. Whereas red means investigate. Investigate now, go back, perhaps take another sample, go across the field. Is there anything obvious that is a problem? It doesn't mean to say there is a problem, but it just means there's something that's flagged up here that's not normal, for example. So you need to go and investigate further. So as I say, visual soil evaluation of soil structure. This is the VES scorecard. It basically involves taking out a cube of soil, to a 20 by 20 centimetre cube of soil, and you lay it out. And first of all, you look to see if you've got any obvious layering, any obvious signs of compaction. If you have, you need to score those layers separately. If you haven't, you just score it as a whole. And then you proceed to break up the soil and look at the shape and the size of the aggregates, and in particular, how easy they are to break up. So you end up with a score of 1, really nice and friable, breaks up very easy, lots of roots, nice small aggregates, rounded aggregates, right down to 5, which is very compact. So you end up with quite a platey, solid structure, and actually it's really difficult to break up. If you want to see a compact grassland soil, come to the AHDB soil pit, which is in the headland, and actually it's probably scores a 4 in terms of compaction. And then compare that to the Hutchinson's pit, which is in the grassland, which is slightly, it's not on a headland, so you can contrast the differences. And as I say, what we do, if it's got a 1 to 2, it's a green. If it's a 3, it's an amber, or 4 and 5, it's a red. So soil organic matter, it's a tricky one, soil organic matter. Everyone has been trying to come up with the value, the critical value below which you drop and everything falls apart. And there is no critical value below. We can't set a single target for organic matter. But what we can do is say what is normal for your soil type. Recognising that organic matter changes according to clay content and moisture content. So what we can do is say we come up with typical ranges. So this is the relationship. This is carbon, which is interchangeable with organic matter. Its relationship with clay content. And you can see that as the clay content increases, so does the organic matter, and the ranges expand. This is for an arable dry region. That's an arable wet region. And that's grassland. So you can see the ranges shift up. And what we're saying is basically, if you're sort of average or above that range, it's good. If you're average and to the bottom of that range, it's amber. And if you're below the range, it's a red. It doesn't mean to say it's bad, but it means you're atypical. And actually, you could be at a much higher level of organic matter based on your soil type. And that's what I've just said there in terms of the colour coding. So we're testing this approach in 2 ways. Firstly, we're going to long-term experimental sites, and these sites have been selected on the basis that they have key drivers of soil biological functioning, and I'll go into that in a minute. But we're also working with 8 farmer groups across the country, from Scotland down to the southwest. And those farmer groups are taking a much smaller scorecard, not doing so much of the biology at the moment, and testing what we see in practice. And we're also hoping that we might extend the range of practices that we see, because we can't cover everything within the long-term sites in terms of management practices. So here are the long-term sites. As I said, we've got 3 sites, Harper University, ADAS Gleethorpe and Terrington. They are looking at the food source. So we've got a range of different organic materials that have been applied for a number of years compared to inorganic fertiliser control. So we're looking at the amount of food and the quality of that food. Then at GW19, we've ploughed up some strips of a 8-year no-till. So we can look at the tillage effects. At Boxworth, we've got drained versus poorly drained sites, experiments. And at SRUC Cravestone up in Aberdeen, there's some really long-term experiments that have got different crop rotations, either with different nutrition or different pH levels. So we're trying to look at the food source, the physical environment, and the chemical environment for the biology, and test the scorecard, but also look at some of the more emerging measures of soil biology. So in terms of when we do the assessments, we've selected for an autumn timing when the soils are moist, although we had to wait quite a long time this year to do it. And we basically take a bulk sample to send off to the labs. So if that was in a farmer's field, we basically select a representative area. We georeference where we take the sample, and then within a 10-20 metre radius of that, we take a bulk sample of soil, topsoil. That would go off for chemistry. We then take 3 Fes assessments, so 3 little pits, and in those pits we'd also count all the earthworms. Bulk density, penetration resistance, but also, as I say, at the experimental sites, we're doing a lot more on the biology. So we're looking at counting numbers of mesofauna and nematodes under the microscope, in order to compare that with some of the DNA approaches that you'll hear about in a minute. So I'm just going to select a couple of the sites that we've been looking at. This is the site at Harper University, which was established probably in the early 90s. So it's got a control treatment which just receives NPK fertiliser. That compared to, at the outset, cattle FYM and cattle slurry. So 23 years of that. And as you can see, this is the organic matter loading. So over those 23 years, almost 130 tonnes of organic matter has been applied, compared to 50 odd from the slurry. And then as different materials are being recycled back to land, over the years we've added in different organic materials. So we've got green compost that have been applied for 13 years. And then most recently, under RAP funding, as a green food compost and a food-based digestate. So you can see we've got different organic materials, so different qualities of organic matter going in, as well as different amounts. And I've just said, it's not just organic matter. We do make sure that we get good crop growth on all the plots. So we analyse the manures and we balance the fertilisers. So this is an example of the scorecard, a very simple scorecard, taking the measures that we're working with farmers with. So according to the different treatments, and I've coloured it up basically. So soil organic matter. This is a sandy loam soil. It's a light soil. So actually, where we've not applied organic matter for 20 odd years, we're actually at the borderline in terms of organic matter. And all organic materials have increased that organic matter, but notably it's the FYM for 23 years and the green compost that have actually increased organic matter to almost the same amount. So even though we've applied 130 odd tonnes of farmyard manure organic matter and only 60 odd tonnes of green compost organic matter, the increase is pretty much the same. Largely because compost is a very stable source of organic matter and actually has already had the breakdown in the heap, whereas the FYM is actually breaking down within the soil. pH, again, is fine. It's a little bit borderline. But again, organic materials do tend to have a liming value. The extractable phosphorus, this site is inherently high. So from a crop productivity perspective, it's fine. But we flagged it up from an environmental perspective. Actually, because we've got high indices here, we need to then go out and see, OK, is there a risk to the environment from this site? If there's no connectivity to watercourses or it's flats, then that's not a problem. But it's just flagging it up. And actually, you may want to think of boosting your organic matter in other ways that perhaps don't supply as much nutrients. And so you can see as we go down, the VES score's fine, the earthworms are fine. So it's just a visual representation of how you might want to score your soils. And I've just put down here also, in terms of some of the other measurements we've looked at, bulk density was reduced where we had those long-term, where we had the highest organic matter on the FYM and the green compost. Interestingly, microbial biomass was only up on the farmyard manure plot, not the green compost, even though they've got the same amount of organic matter. And again, that's probably due to the stability of the compost going in, whereas the farmyard manure needs some processing and is actually decomposing, so it does actually boost the biomass relatively higher than the compost would. If we take a second site, this is up at Aberdeen. This is the very long-term site. It was established in 1961. And it's a long-term 8-course rotation. So we've got root crops in there, we've got cereals, we've got 3 years of grass. And they are present every year. So you can see that the different crops are present as you go across the field. And then as you go that way, you've got different pH levels manipulated with lime or sulphur. So we've got 4.5 up to 7.5. So a whole range of different conditions in terms of what residues are going in, what cultivations are done, but also what the pH is like. So a good test bed for certainly for soil biology. We couldn't go into every single plot. There was lots of plots. So actually we just chose 4 pHs, and this is the scorecard as an average across 4 different crop types. We have got them for separates, but I'll just show you this as a demonstration. So and I've put some different measures on here that we're doing. Just to check the pH, yes, 4.5 is around about 4.5, 7.5 is 7.5. The organic matter, this is Scotland, so a higher organic matter content. It's still a light soil, but a high organic matter content. And the soil structure is fine. But when we come to the biology, so this is microbial biomass carbon, you can see actually there's almost like a bell-shaped response. A red flag, this is using international benchmarks, so we're still working out whether they're appropriate for UK soils, but they do give a red flag for low pH, which it's not surprised, but they also give an amber around 6 and 7.5, so it's quite encouraging that liming to 6.5 is optimum in terms of microbial biomass. This respiration is the CO2 burst method. I've not coloured it up because I don't feel confident at the moment with the benchmark data that we've got. But interestingly, it follows the biomass. So it's low here and at the far end, with the optimum being 6.5, which is encouraging. Earthworms are pretty bad, but again, that's an average across. So we've got we've got root crops in there as well. So it looks different if you separate out the different crop types. But just as an example of how we're developing and how it might work. So finally, before I hand over to John, I just want to talk about biological indicators. As I say, a separate aspect of the programme is to try and extend our understanding of soil biology. And at the experimental sites, we're taking a whole range of other measures. And I'll just give an example of what we're doing with nematodes and whether we can develop some sort of framework to interpret what we see in terms of nematodes. So nematodes are routinely assessed in, say, root crops because they're plant pests and they're transmitters of viruses. So they are measured already, but the plant pests are only one aspect of the nematode community. There's a whole range of different nematodes that actually sit on various stages of the food web, so different levels within the food web. So there are, as well as the plant feeders, bacterial feeders, there's fungal feeders, there's those that eat the others, and there's those that, omnivores, they eat whatever. So actually, by looking at the whole community, can we use that as an indicator of the overall health of the soil biology? And there's been quite a bit of work actually in the scientific community to try and do that and come up with some different indices. And actually there's 2 indices that they've developed. One's called an enrichment index and one's called a structure index. The enrichment index basically pulls out those nematodes that respond to high nutrient sources. They rush in, use all the nutrients, and then so they've got a short life cycle. Whereas a structure index basically indicates those nematodes that have more a longer life cycle, and they reflect the whole stability. So disturbance is an issue for them. And we can basically plot these indices against each other, and depending where you are, you can tell a little bit about the community. So if you're down in this quadrant here, it's a stress community. It's depleted in nitrogen, and it's very unstable. Compared to over here, which is a highly stable community, but it is a little bit N-depleted. And then the N enrichment goes up that way. So we've actually tried to colour this up with our flags. And this, as I say, this is very early stages, so we don't know whether this is right or not. But we're suggesting that actually you really want to be over here in terms of your nematode communities. So if we look at the Harper Adams site with regards to that, here we go. Here's all the different treatments. Actually, most of the organic materials do score a green. Our control is an amber, and our slurry is a bit too enriched. It's an amber. So it's sort of going in the right direction. And as I say, we're hoping to try and progress this further as we get more and more data in across over the project. But one of the issues is actually it involves a lot of work. You're basically, with a microscope, counting what you see and identifying what you see. So it's laborious, and it will inevitably, as a result, be costly. So are there other ways in which we could potentially be looking at this? So that's just a summary of what I've just been talking, but actually that sort of leads into what John is going to be talking about, as whether we can use molecular techniques to identify these organisms rather than just looking at things under the microscope. So I'm going to hand over to John now, but while I do, if there's any points of clarification, we'll take some questions at the end and have a bit open up for a discussion, but if there's any specific clarifications of what I've just said, otherwise I'll take some questions at the end. Yeah.
The Harper trial with digestate.
Yeah.
Can you tell us whether that's separated or?
It's whole. Yeah. Yeah.
How did you measure the
Biomass. Yeah, so that's it's a chloroform incubation. So basically, you analyse the carbon content of the soil, and then you kill everything, and you analyse the carbon content at the end, basically. Yeah. Okay, I'll hand over to John.
Morning, everybody. So my job, if I can get it to work, is to talk about some of the research we've been doing within the Soil Health, Soil Biology Partnership up at Fera in Sandhutton in the laboratory, trying to use some of the more recent techniques to look at the biology of the soils in the trial samples that Anne's been talking about. Traditionally, of course, this has been done by people like me with their Petri plates and a myriad of different microbiological media. Those techniques really look at the tip of the iceberg in that all you're going to get on your plates are the most predominant organisms and the fastest growing organisms that then outcompete everything else. The other issue is that a lot of what the microbiology in the soil, a lot of those organisms simply don't grow in the laboratory on artificial media. So how do we get around that? Well, we can look at the soil DNA. Every organism has DNA that is unique to itself. So we've used two different methods to do this. First of all, we need to extract the DNA. So the first year of the project, we've been pretty much looking at all the different methods that we can have at our disposal to isolate DNA from soil, which is quite a difficult thing to do, and then purify it free from all the humic acids and things that interfere with the methods that we're applying. Once we've managed to do that, and we think we're at a point now where we've got some methods which are pretty reliable, so we're taking it forward. We're using two different techniques. Firstly, quantitative qPCR. This is a procedure which allows us to quantify individual organisms for which there are known unique DNA markers. So by measuring the amount of that DNA marker that's specific for a particular organism, we can get an estimate of how much of it is in a soil sample. The second one is now becoming a much more useful tool, which is high throughput DNA sequencing. So in this case, instead of just looking for the marker, we look for the marker and then we sequence it. And the sequences are unique to each individual species that's present in the soil. And using fancy bioinformatics and computerized systems, we can actually start to tease out all the different organisms that are present in the soil from a sample of the DNA. So the first method, the qPCR method, on the left is just a list of all the different organisms that we currently have these specific markers for. Most of them are plant pathogens because that's what we've been worried about in the past. We're trying to increase that list to include organisms that are beneficial in the soil as well. So we're not just looking for the bad news, we're looking for the good news organisms as well. What we've been doing so far is just validating all of these tests. You can see that if we graph the amount of DNA of each organism against the test result, we get a nice linear relationship so that we can measure amounts. The bottom bar charts are where we've actually added target organisms into soil in known amounts in either low, medium or high levels, so you get the three different size peaks. And what you can see from these is that if you do this with different soils, occasionally you'll get a problematic soil, in this case soil number 12, which was very high in organic matter. It was from one of the trials that it had extra organic amendment. So it's not always 100% reliable. We don't always get the DNA out that we want to, but we're looking at methods to improve this all the time. Here's an example of some results that we got from NIAB's STAR trial, where they were looking at the effects of rotations. And here we have a spring or a winter break crop compared with continuous wheat or alternate fallow and wheat. And here we're looking at the amounts of the take-all fungus in the soil. And you can see that the break crops are having an effect at suppressing the take-all. We've got a PhD sponsored through this project. Here's Emma, and she's looking at trying to relate the levels of DNA of soil-borne pathogens to the risks of crop disease. So, okay, you get a result, you get an amount of DNA, but what does that mean to the grower in terms of the risk of a disease happening in the field? And Emma's looking at various model systems. So she's got an onion trial, looking at white rot. She's got verticillium wilt in raspberry, fusarium basal rot in daffodils, and then the data you just saw in the take-all in wheat. So looking at trying to monitor the levels of DNA and then monitoring the risk, the amount of disease and modeling that to see if we can get some predictions. On top of that, we've got within the project, we've imposed treatments. So we're looking at organic amendments and how that might affect pathogen survival over time. We're looking at the effects of biological agents added into the soil, so things like Prestop, Glyocladium, and arbuscular mycorrhizal fungi. What happens if we add them in to the pathogen populations? Is the competition? Does the improvement in plant growth help to defend the plants from diseases? So lots going on in that PhD. Second PhD is using the other technique I mentioned, the high throughput sequencing, and a process called metabarcoding. So every organism has its own unique barcode, and we can extract the DNA, sample the soil. We're using kilo samples from multiple sites, and then mixing that very well, and then taking 10 gram subsamples from that for the test. We're using an extraction method called PowerMax, which is a commercial kit, but it's working quite well in the hands. And we're looking at these different soil management treatments in the trials and described. So looking at different soil types, different pH levels, organic matter, different cultivations, cropping patterns, and plus and minus drainage. So then once we've got the DNA extracted, we do the metabarcoding. So this involves a PCR step. We amplify the target genes and then use the high throughput sequencing to get basically millions of sequences from single soil samples. Then what do we do with those? Then we put those to our bioinformaticians, and they use software, in this case called CHIME2, as a bioinformatics pipeline. You put all the sequences in one end, and it does some fancy analyses and tells you how diverse the soil is in terms of bacteria and fungi. And using different databases of DNA sequences, you can actually identify to certain levels, certain taxonomic levels, what those bacteria are and what those fungi are in the soil. So this is what results start to look like when we come out. We get these really psychedelic bar charts, which we ponder over for days. But what you're seeing here is analysis of soil from the Gravestone site with the different pH levels, 4.5 to 7.5. And I think you can probably see at the extreme level here, the samples that are coming from the 4.5 look a bit different from the others. And similarly with the fungal. So bacteria and fungi, something's happening in terms of the diversity in the soil. So to explain what this is, each bar in this diagram represents 100% of what's in there, and then the individual colors are the different sequence types that you can find. So at the top you have the bigger bars, the more abundant species that are in there. Going right down to the bottom, then you've got literally thousands of different organisms down here that are in low abundance. So we can weed that out, and we can identify perhaps the top 10. What are the major part of those communities? For bacteria, in case of fungi, this is at the level of phylum, so this is high up in the taxonomic tree. We're only getting 3 different fungal phyla. And then we can do fancy analysis of the diversity. So these are measurements of how diverse a community is. And you can't see the details here, but this is comparing from the Gravestone site again, comparing each pH level with the other pH levels. And we see that even in pH, between pH 4.5 and the other pHs we knew was different, between the other pHs, they're all significantly different compositions of bacteria and fungi present. So the pH is having an effect on the communities. Interestingly, we looked at a single field and how diverse, if you took multiple samples from a single field, how diverse is that? Here we had an onion field in the previous crop that was now under either under cover crop or not. And we didn't see, when we did the analysis, we didn't see significant differences across all the samples that we took. So it seems that at least for bacteria and fungi, the communities that we find in a particular field are quite stable wherever we take the sample.
When you took the sample for that?
So this one would have been taken in the springtime, in about March, something like that. Sorry, this was taken in November. There was cover crop, but it was only partially established. Yeah. A few things to beware of. The methodology is very sensitive, and if you take at each stage of the methodology, there are various biases that come in. And for example, when we were analysing the samples from the Harper Adams trial site, we actually used 3 different DNA extraction methods. So one was the method routinely used up in Scotland at SRUC. One was used by a colleague also working on this project at Lincoln University, Matt Goddard. And the other one was the method that we're using at Fera. And what you see here is that the samples cluster according to the DNA extraction method that you've used and not according to the type of organic amendment that we applied. So the differences between methodology are more visual than the differences between the actual treatments that we're trying to study. So that means that we have to be very standardized in the way we do this. If we want to compare one sample with another, then we have to really be careful that we're using the same method throughout. We've looked at this is all measurements involving the diversity of the communities. But what happens if we use the qPCR to look at amounts of DNA? And here we have 2 probes. One that is specific for all bacteria, so it's not just for a single species, it's for any bacteria, ribosomal RNA that's only found in bacteria, and again ribosomal RNA that's only found in fungi. And we could see here that there were at least some differences in amounts of DNA, particularly where farmyard manure had been applied over many years. The lower levels in this case actually refer to higher amounts of DNA. So higher levels of fungi and bacteria were recorded in the Harper Adams samples with farmyard manure. So we're applying this now at Fera. We have started something called the Big Soil Community, and this is a group of interested farmers working with ourselves and Matt Goddard at Lincoln University to generate data that will give us a baseline of what's out there. So what we're doing is we're asking the growers that are involved in this community approach to send in samples in the autumn time. We then do this metabarcoding analysis on the samples, and we send out reports in after Christmas. And this gives an opportunity to understand the range of different levels of soil diversity in terms of the organisms that we're finding. So it's a community. We've got about 200 samples in the first year, 200 members of the community now. We're performing this metagenomic metabarcoding analysis. We're providing reports both as individual reports, so what's present in your soil, and also a benchmark report which shows you where your soil sits in the range of diversity across all of the samples that we've analysed. This is what a sort of mock-up type report would look like. So we say the total number of sequences that we're finding, so over 1 million reads for bacteria, 764,000 for fungi. We tell the most abundant organisms that are present at all the different taxonomic levels, down from phylum right down to species. And we give a list of where we can identify those organisms, whether they are not already known to be detrimental or beneficial in some way or another. We are then provide the benchmark report like this. So this is the range of diversity that you might see, and this is where this individual sample sits in that range. And we're asking each grower to send in metadata that accompanies the sample. So we're getting data on the pH and the texture, the current cropping history, the amount of organic matter that's in the soil, and some data on pesticide usage. At the moment, it's fairly simple data. Is it used or not? But that's going to develop further in the future. So this is where we are at the moment. We're hoping to grow this this year to 500 members. It's a sort of crowdsourcing effort. So it's very much at the experimental stage. It's not a commercial test by any means, but it's allowing us to build a picture of what kind of diversity is out there in the soils that we have and relate it to some of these factors. We're extending it into forestry this year as well, so it won't only be, most of the samples we've had have been arable samples, but we're going to look at forestry and more grassland samples. And we're also hopefully this year introducing a third test. So we've been looking at fungi and bacteria up to now. We're going to start looking at nematodes and other mesofauna as a third option. And I think that's all I had to say, so I'll stop there and we can open it up for questions. Thank you.